We are going to start talking about the muscular system by reviewing sarcomeres. So here is our topic and outcomes for the day. And so just to start off, here's an overview of a muscle cell. Excuse me. The important parts that we've got on this are as follows. So we've got the sarcolemma, which is just the cell membrane or the plasma membrane of a muscle cell. The sarcoplasmic reticulum, which is basically a storage site for calcium, which is important, hopefully, from what you remember from your physiology class. We've got the terminal cisternae, which is actually where the calcium is stored in the sarcoplasmic reticulum. And then we've got the T-tubule, which is these holes or invaginations is often how they're referred to, where an action potential can travel deep down into a muscle cell. Now, when it comes to the myofibrils, there's actually 6 proteins that are involved. We've got 2 that are contractile, actin and myosin, and actually there's a third one called titin, which you've probably heard about, but you haven't ever heard of it referred to as a contractile protein. And there's actually going to be a lecture on that a little bit later. So titin is also contractile. Regulatory, we've got troponin and tropomyosin. And then structural, there's titin where you expect it, as well as nebulin. So when it comes to these proteins, you've got myosin, which is attached to the M line. It moves actin. It's referred to as a thick filament, whereas actin is attached to the Z discs. It's a thin filament, and it's pulled towards the M line by myosin via the cross-bridge cycling. Tropomyosin is the blocker. In other words, it prevents actin and myosin from getting together. And then you've got troponin, which ultimately will move tropomyosin out of the way. And it can attach to calcium, actin, and tropomyosin. Now, within a sarcomere, there are several bands and zones and lines and that sort of thing. And so just to start off, we've got the Z line. Okay, and we can see that here and here. And the Z lines are just or Z discs are just the end of the sarcomere. One sarcomere goes from one Z line to the other. And this is where actin attaches. And it's not shown on this picture here, but myosin actually connected to the Z line via titin. Okay, and titin's very elastic, and we'll get into that a little bit later. And then you've got the M line, which is the middle of the sarcomere, which you've got here, and that's where myosin is attached. Now, getting into the specific bands, you've got the A band, which is here, okay, and that is where you have myosin. If there's myosin, it's part of the A band. And so there is some actin there, but you've got all of the myosin. The H zone, which is shown here, okay, that's where you have some myosin, but no actin. Okay, so that's only myosin. And then finally, you've got the I band, which is kind of the actin version of the H zone. So the I band is where we have only actin, but no myosin. And the reason that they're called the A band and the I band is A stands for anisotropic. And what we mean by that is that light can't get through. So when we look at them through a microscope, they're very dark. So the A band is dark, whereas the I band is light because it's isotropic, or in other words, light can get through. Now, as you look at this figure, you can see that these bands and zones change during muscle contraction. At least some of them do. The A band stays the same because the myosin filaments don't get any shorter. They're just constant. All they do is they pull the actin towards them. And then we've got the H zone, which gets shorter because we've got more overlap. And as we've got more overlap, there's less area where there's only myosin. And then the same is also true of the I band. As we have more overlap, there's less overlap that actin has, or excuse me, there's more overlap that actin has with myosin. So for this lecture, here are 3 things that hopefully you can do. Can you define a sarcomere? In other words, how do we determine what is a sarcomere? Can you classify those proteins in a muscle as either structural, contractile, and or regulatory? And then finally, can you describe what happens to the bands and zones of a sarcomere during muscle contraction?